so many interesting talks from all across Africa, and we've got an awesome lineup again this evening. We're going to jump right in, and our first speaker is Craig Peter, and he's an associate professor in the botany department at Rhodes University. And one of his big passions in life is orchids, which is one of the main reasons why we started LepiMAP in the virtual museum. And if you've ever seen any of his talks on orchids, it's absolutely jaw-dropping amazing. But this evening he's going to talk to us about LepiMAP and how he's been using LepiMAP data to better understand butterfly pollination in South Africa. So over to you, Craig.
Thank you very much. Hello, everyone. Good to see some old faces I haven't seen for quite a while. Megan's mentioned LepiMAP, but in fact, my time with all of this came via LepiMAP. So LepiMAP was my first contact with citizen science. And I've been quite an avid LepiMapper since then. And exploring LepiMAP really led to the idea for OrchidMap. But I just want to tell you a little bit about what I've been getting up to with LepiMAP in the last couple of years, trying to figure out what we can do with the records in terms of pollination. So not even worrying about orchids, my real entry base level research is pollination. And having been interested in butterflies since I was tiny, obviously the attraction all along has been to do some work with butterflies. So the things that have really caught my eye obviously are red flowers, which is a bit weird because I'm red-green colorblind, so I don't really even see the red flowers all that well. But I've been really interested in trying to figure out what's going on with these bright red flowers, given that most insects don't see red very well, and most red flowers are bird-pollinated flowers. So one of the plants we've been working on is Pelargonium inquinans. So this is what we're looking at here. And it's pollinated by various pyrrids and big swallowtails. So the main pollinators probably are Papilio demodicus, and that's pretty much it down here in the Eastern Cape. And these photos, these obviously are my photos working in the field, but LepiMAP proved to be a really great source of additional records from around the country. So that sort of whet my appetite to see, can we mine more observations than I was able to generate just on my own bat. This isn't a great photo, but it's nice because you can see these little orange bits of dribbles around the butterfly's proboscis, which is the pollen. So this pelargonium has this bright orange pollen, so it has this nice stain on the butterfly, which gives away that the butterfly has visited this pelargonium. One of the other things I've been really excited about is Bauhinia galpini, pride of the carp. And that's up in the Lowveld, and it's very exciting because it puts its pollen on the wings of the butterflies, which is a pretty unusual place to put pollen. If you think about insect wings, they're flappy things, they're beating around, and so it's not necessarily the best place to put pollen. So I used that as an excuse to go up to the Lowveld a couple of times, and it seems that only the big swallowtails are the legitimate pollinators. The pyrrids all do something completely different. They all sit on the outside of the flower, and they probe their proboscides between the petals down into the nectar tube. There's the nectar tube down there, And so these guys are robbing nectar from the flowers, and very rarely make contact with the reproductive structures of the flowers. There are the anthers up there and the stigma. And so this butterfly is stealing nectar and not doing anything for the flower. So that research showed very nicely that there's just a handful of butterflies that really visit the flowers en masse. Some of the big swallowtails and Catopsula and a few other whites. But you can see the number of visits that are recorded for these three species. But then when I scored whether or not those visits were those legitimate visits going into the flower, or the butterfly sitting on the outside of the flower, it changes quite dramatically. So you can see that even though these things are visiting the flowers really frequently, they are not likely to be pollinating the flowers at all. And then the final one, which has just been published at the start of this year, one of the big excitements at the start of lockdown was getting this paper out looking at Gloriosa superba, which is really just the most spectacular story all around. And I could speak for 40 minutes at least just on this. So alas, I'm just going to skip through the highlights. It's pollinated almost exclusively by Aronia cleodora down here in South Africa. It's mainly a coastal species through the Eastern Cape and KwaZulu-Natal, and then it goes inland through Mpumalanga and Limpopo into tropical Africa. In South Africa, just about exclusively by Aronia cleodora, as I said, and the butterflies arrive and land on the outside of the flower. This is just the most bizarre and amazing flower. It's an inside-out lily. So the petals are recurved and pulled outside, and there's the pistil in the middle and the stamens radiating around that. And the butterflies arrive and land between adjacent stamens, and the anthers rub on the underside of the wings and deposit the pollen on the wings, a bit like the Bauhinia. So there you can see all this massive smears of pollen on mostly on the hind wing of this butterfly. So there's loads and loads I would love to tell you about, but there's not really time for that. But the link of the Gloriosa study was that mining LepiMAP, going through all the pyrid and papilio records in LepiMAP, turned up a whole suite of other observations of pyrids visiting Gloriosa superba at various places throughout Africa. And it really reinforced what we were trying to say about Gloriosa superba down here at the very southern tip of its range. Gloriosa superba goes all the way up through Africa and in fact into India. And so these records really were able to strengthen our story quite substantially. So that got me thinking. I thought I should be doing this in a more organized way. And so I started a little bit haphazardly at first, but I started screening all the records and trying to identify and score a few traits about each of the flowers that the butterflies in the LepiMAP records were interacting with. So in a nutshell, what I did was I started at the beginning. So here's the very first LepiMAP record of a butterfly interacting with a flower. And pretty much all I did was copy the URL for the record. And from that, I can extract the LepiMAP number. And of course, with the amazing services of Renee, I can just send him a list of these LepiMAP numbers, and he sends me back all the gory details of each of those records. So from my perspective, I'm really interested in the identifications of the butterflies. So all this data can then just go straight into the database. At the same time as recording the URL, I also try to figure out what what the plant is that the butterfly is visiting. So here it's pretty clear that it's a daisy, so I know what family it is. And with a bit of thumbsuck and guesses, I think it might be an Arctotheca. I try to get it as close to the species as possible, but if it's just a genus, that's also works. And obviously the family of the plant. And then I've been scoring the colors of the flowers. So I've got a list of obvious colors, and I've taken this a step further more recently, trying to measure, get a little bit more quantitative about the colors on the image. So far, going for a good couple of years really, I've managed to get through about half of all the LepiMAP records. So I looked today and there's just over 226,000 records. And unfortunately, not having done anything, I was just a little bit ahead of halfway, and now I'm slipped back almost to exactly halfway. So whenever I get a little bit of enthusiasm, I do another few thousand. And all of this has yielded 21,000 observations of a butterfly interacting with a flower. And with the interest in the color and really trying to think about or look at the preferences of butterflies to different colors. I'll just show you some rather crude data from all these records. These are just all the records just lumped in as one. So all butterflies and moths visiting flowers. And you can see, as I said at the start, I'm interested in red. So not a lot of red flowers visited by butterflies. Yellow, pale blue, pink, and white are the colors that are attractive to butterflies. And then we can start to untangle those by some of the different families. So the Lycinids, the Pyrids, the red starts to become a bit more commonly visited, red and purple. And then rather satisfyingly, in the Papilionids, red and purple get close to making up a quarter of all the visits of the butterflies. Nymphalids, rather few, and it's really just Eripetes pretty much that is dominating this segment of red visitation. So finally, looking at the colors, this shows that the Pyrids and the Papilionids visit red flowers significantly more than predicted if we just went with that average red visitation across all the butterflies. So in comparison to that average number of visits to red flowers by all butterflies, swallowtails and whites visit red quite a lot more. Of course, there's all sorts of other things to do. I've only just started dabbling with this. There's 2 third years who have had to change their projects this year because of COVID, and they've been experimenting and trying to get to grips with some of the ideas. So we've started making these network connections between, so these are just mainly butterfly families and a few day-flying hawk moths and a few other moths, and trying to link those up to the different families of plants. So you can see these are by far the most important families that butterflies get nectar from. Daisies and Verbenaceae, acanths, some of the milkweeds and so on, and then lots of other little things. We're also interested to try and figure out, well, to see the importance of invader plants. So all the things in white are weeds, basically, and you can see that things like verbena and lantana and tridax are really heavily utilized as nectar sources by butterflies. The things in green and Bauhinia galpani, these are the top, I don't know, 20 so species in the data set. So finally, just to say, if you are LepiMapping, rather than tightly crop your photo around the butterfly, please consider cropping your images a bit wider, because there's all sorts of things that we can get by being able to see the flowers nicely. And if you're feeling particularly generous, you could throw in a second photo of the flower that it was on. And if you know what the flower is, you could put a note in about the species, the genus, even just the family. I've become incredibly good at it, identifying weird and wonderful plants from just the tiniest little scraps and of evidence. But if you can make that process a little bit easier, if you know the plant, please put it in the notes. It will really help with things I'm not familiar with. And then very finally, the final thing I want to say is the same applies for birds. If you're taking photos of birds and you see birds interacting with flowers, obviously the sunbirds and the sugarbirds are great, but we're really interested in all the other birds that visit flowers, weavers and all those kind of things, orioles, and there's tons of birds that visit flowers, and some flowers are specially adapted for non-sunbirds and non-sugarbird pollination, like this Allopharax. So this Allopharax is better pollinated by weavers than malachites. So if you're taking photos of birds, please include the flowers as well. All right, that's what I've got to say. Sorry if I don't know, I rushed through that a bit. I hope I didn't go over time too much.
No, well, thank you, Craig. That was absolutely amazing. It's such an awesome example of what citizen science data, what we can do with it. And I think we should definitely get the message out to our VM contributors that since they can put 3 photos in a record, that if they photographed a bird or a butterfly or something on a flower, then, add a photo of the flower to that record, or of the plant that it's on. It's definitely very, very cool.
Thank you. Yeah, I've even written a little bit of a sort of outline of what would be nice things how to do it, basically. So.
Yes, well, you can send that to us, and we can definitely spread the word and let people know. Yeah. I see we have one question. Steve Woodall asked, is there anything about depth of flowers visited by butterflies and how it correlates with their size?
Yeah, so I've been frantically measuring tongue lengths of butterflies. And one of the things that I haven't been scoring all that diligently is how much longer the tongue is relative to the tube or the depth of the flower. So like this example here, the butterfly's tongue is only just getting into the opening of that very short tube of the stachys. So you wouldn't expect this to be a very efficient pollinator of this. I'm sure this stachys is adapted for bee pollination, and this mocker is really just stealing nectar. So there's a lot more that can be siphoned out of all this data, but that's in the early stages.
Yeah, gee, it's fascinating. Well, we'd be happy to help in any way that we can with the projects in the virtual museum.
Cool.
Yeah, it's very exciting. Thank you, Craig.